Jana Bagci.
Thank you. Hi, everyone. Welcome to my talk titled BGC Atlas, a web resource for exploring the diversity of biosynthetic gene classes in metagenomes. So, although there have been a few talks already in this session about secondary metabolites and nature products, I'd like to start with a brief introduction to what they are. So, when we talk about secondary or specialized metabolites or nature products, as they are also called, these are compounds that the organisms produce, but they're not directly involved in their growth or development, but they typically have important ecological or physical advantages to those who produce them. And the production of these molecules, or the products, are often driven by the so-called biosynthetic gene clusters, which are groups of genes that are physically linked together and collectively drive the synthesis of molecules. As an example shown here for the product called neocati casinostatin biosynthetic gene cluster from MIBig. As other organisms, microbes also produce these molecules, and they produce a diverse range of them with a wide array of functions and structures. And these molecules that the microbes produce, the secondary metabolites or the nature products, have many applications in such as medicine, biotechnology, or agriculture. They are the sources of many antibiotics or anti-cancer drugs that we use today. They're also used as immune suppressant, or they also have plants to grow as a plant growth promoters, and they also play roles in interspecies communication as agents in communication between microbes themselves, but also between a microbe and its host. But although that there have been many efforts to characterize these products and the gene clusters that are responsible to produce them from the community, most of these efforts have focused on isolate genomes, or at best high-quality metagenome assembled genomes. So using metagenomes to predict these compounds or the gene clusters that produce them has been basically impractical so far, because these regions are quite difficult to assemble because they usually span up to hundreds of kilobytes in length, and the communities or the microbes that are found in communities that produce them are often too complex, making it difficult to be in the context into high quality mix. And this fact has also led to ecological implications of these products being left undiscovered. So one such effort is actually from a colleague of mine who is presenting it tomorrow at poster B182 if you want to see that. But recent developments, both in sequencing technologies such as long-read sequencing and also in the computational analysis of metagenomes, have led to identification and investigation of these products and gene clusters in the recent years, in the, let's say, last 2 or 3 years. And in this work, we want to take advantage of this, and came up with the idea to explore and analyze the diversity of these compounds in a global scale in our work titled BGC Atlas. So this is a resource that's a collection of biosynthetic gene clusters found in environmental samples, so as in metagenomes, that offers exploration and analysis of their diversity, that associates them also with all the metadata that we can collect for each sample, and also lets the users to query for biosynthetic gene cluster that they are interested in, if there's a similar one in the collection that we already have. As an overview, what we do is we collect every publicly available environmental sample that we have access to, the associated metagenome assemblies and the metadata with them. We annotate the assemblies to identify the biosynthetic gene clusters and further cluster them into so-called gene cluster families, which I'll come back to later. And all of this data that we produce and collect from publicly available resources are collected in a central collection, which can be queried and viewed by users through a web interface, but also the users have possibility to search for the gene clusters that they identify themselves in this resource that we have produced. But briefly, the way that gene cluster calling and clustering works is that we take not only the metagenome assembled genomes, but whole metagenome assemblies, so all of the context that are produced from an assembly, because when you bin context into mags, as probably most of you know, that we lose a vast majority of them in the process, especially when we want to work with the high quality ones. So we take everything that's in there. We use anti-smash for gene cluster calling, and we use big size for clustering similar gene clusters into gene cluster families, so GCFs, the so-called GCFs. And these are, so as an example shown here, these are very similar in structure and in sequence, and also in the synteny of the genes and their domains. And these are potentially responsible for the synthesis of same or similar products. But one major problem with metagenomes is that most of the gene clusters that we identify are not complete, but only partial. And for that, what we do here is that we initially cluster only the gene clusters that have been annotated as complete by anti-smash, and then partial ones or the known compounds from databases such as MIBIC are assigned in a second step to the ones that were clustered in the first step, so to the complete ones that were clustered in the first step. And so far, we have analyzed over 30,000 data sets from Magnify, more than 2/3 of them being host-associated and more than half of the samples being from digestive system of mostly humans and mammals. We also have more than 5,000 environmental and 1,000 engineered samples, such as bioreactors. We have identified nearly 2 million gene clusters, only about 10% of them being complete. And of these, more than 150,000 complete Biosynthetic gene clusters, we produced almost 11,000 gene cluster families using the complete ones. And we have also associated metadata for each one of these samples. So as an overview, what we get is from these nearly 2 million Biosynthetic gene clusters, most of them belong to most of them produce the compounds that belong to the category RIP, and most of these RIPs are cyclic lactone autoinducers, which are found mainly in the digestive systems of humans and mammals. But as we dive further to associate the products with the biomes, we notice that there is already at the highest level, like when we compare the environmental samples to host-associated samples, for example, there's already a difference in the type of product that these gene clusters or gene cluster families produce, such as the terpenes being almost exclusively being produced in environmental samples, whereas the cyclic lactone autoinducer being produced almost exclusively in host-associated samples. Same also goes for when we go further into host-associated samples and compare them for different hosts, we also observe differences in the type of products that each microbiome that's associated with a different host can produce. There's even more clear in when the environmental samples are compared, like when the aquatic samples are compared to the terrestrial samples. We see, for example, phosphonates produced only in the almost only in the aquatic samples, and having quite a few members that come from terrestrial samples. But this is just at the Biosynthetic gene cluster level. Then when we look at the gene cluster families, so the groups of Biosynthetic gene clusters that are supposedly responsible for producing the same or similar compounds, we see even more specificity, such as when we look into the biggest gene cluster family that we have in our database at the moment, that has almost 17,500 unique Biosynthetic gene clusters across all samples. 90% of these are host associated. But this gets even more dramatic when we look at a few other examples, such as one we have here. And then our PS compound has 95 occurrences, 95 gene clusters in all the samples that we've collected. And all of these are found exclusively in human sequence samples, and not even anywhere else in the human body. Another example is an arylpolyene which is found, which has 103 occurrences in our collection at the moment, and found exclusively in mammalian feces samples, and also not in humans. So not in human feces samples, but only in mammalian feces samples. So looking at this at a global level, because so to quantify the habitat specificity of the GCFs, we see that 76% of GCFs that have at least 5 members are found exclusively in one specific habitat at the highest level. So when, for example, environmental versus host-associated versus engineered is compared, this is still the same at the second level of the hierarchy for biomes, and still, even at the deepest level, at the fourth level of the hierarchy, like, for example, when skin samples versus gut samples are compared, we see that more than 50% of the gene cluster families are habitat specific. And with so much data also comes a lot of computational problems. I mentioned one of these here that we're trying to also solve. One of them being the partial gene cluster assignment that I mentioned earlier, and the other being allowing users to query to search similar BGCs that are deposited in our resource. So one thing that we offer is to use the big slices query mode. There's big slice is a feature-based algorithm based on domain similarity of the sequences, but we're also developing an alternative method to identify gene clusters that are partial or that are submitted by users to be queried in our database based on domain similarity or the sequence-based domain similarity of the gene cluster families. What we do is we first identify all the functional domains on each gene cluster, because the domains are most of the time what's responsible for producing the type of the product. Then we do a all versus all comparison of these domain structure, let's say, based on the shared domains, the similarity of domains with each other, but also we score, for example, some domains better, higher than others, because they are the core domains of a gene cluster, and they're based on their localization in the cluster itself. And we use this to cluster complete BGCs, but also assign partial or user query BGCs using a pairwise alignment of domains in this case instead of sequences, because as I've said, we think that domains are the functional units that actually matter for the production of the kind of the secondary metabolite. So as an outlook, the tool is already available at the website shown here. The search function will be available in a week or two, and we think that using this, users can explore the diversity of environmental samples, and we also allow clustering and search of BGCs found in environmental samples, which can help uncovering the ecological and evolutionary factors that influence the biosynthetic potential of microbial communities. So with that, I would like to thank my colleagues, my funding agencies, and you for listening. And for more discussion, you can come to my poster in the evening. Thank you.
Okay, we may have time for one question.
The gene cluster family are very specific to like location and the host. I know also the species are also very like location specific. How do you see the sort of association between gene cluster family with species in terms different?
That we haven't explored yet, mainly because of computational resources. So we have focused on BGCs and GCFs, but haven't done any taxonomic analysis yet. But that's planned. That I cannot say if there's a species or whatever taxonomy specificity for GCFs or what level it is at. I suspect there is a lot, but I don't have numbers at the moment. But thank you.